So I'm going to talk about something a little bit different today. Did any of you read the paper? Who read the paper?
Daddy.
So there was a few of you. Okay. So it was about salt. Salt toxicity. Why do we worry about salt a lot in Canada? What do we use salt for in Canada? Anybody know? Some problem you don't have here. We have freezing roads, and we have to put salt, a lot of salt, on the roads in winter to get rid of the ice so it's safe for cars to drive. But that's actually becoming a major environmental problem because we put so much salt on the roads and it runs into the streams and the ponds and causes toxicity. So that's why I sent out that paper for you, just to see how that's a specific problem with salt in Canada. One you're never going to have here. But you will have problems, and you are having problems, I think, with salt toxicity in Brazil. So that's my theme today. So my title here is a pun. What's the Portuguese word for pun, Dal? You know pun? A joke.
Puss.
Puss. Puss.
A joke?
A joke. It's a joke. It's like you use a word for a different meaning.
Okay. Una piada.
Okay, yeah. Okay, so I'm going to be talking about the potential for salt toxicity, and that's where the joke is. And it's about using physiology to address a major environmental problem. And a number of people have contributed to this project already, and I'd like to highlight Beverly, my postdoc, Beverly Hoyt-Keepo, who has not been here but hopes to come here sometime. Danielle MacDonald and Martin Grissel at Miami. A US EPA scientist, David Mount. William Adams, who now has his own consulting company called Redcap, but he used to be the head of research for a big metal corporation called Rio Tinto. And Kevin Bricks, who has been here. He was here last year. He's been here twice, actually. He's been on the expedition as well. And in fact, it's Kevin that really got me into this whole project. So, just to highlight these two, Kevin Bricks, who now owns and is the principal director of his own consulting company in Miami called EcoTox. Many of the experiments you're going to see we did together, and he got me interested in this whole thing. And now we have some funding at UBC, and my postdoc, Bev Poe, is carrying on this work at UBC. So, a little bit of background. So some of you read the paper about the problems of salt with rainbow trout, but there's all sorts of problems now with salt in the environment. And it's not just sodium chloride. It's all types of salt. So the most serious problem right now is from coal mining. In coal mining, the sort of mining they do in the eastern United States, they basically just take off the top of a mountain and grind it up to get the coal. And they discharge vast amounts of water, which contains a lot of different salts. And that goes directly into rivers. And in fact, since President Trump was elected, there's now no restriction at all on what they can dump into the rivers. There used to be restrictions, but no longer. Hard rock mining also has effluent. Trona mining, that's mining of sodium bicarbonate, has a lot of effluent. Now we come to some potential Brazilian problems, such as oil shale water and deep oil exploration process water, where the water that comes back out of the pipe is very salty. Poor agricultural practices result in salinization of rivers. In Canada, road salting. And throughout the world now, fracking, basically where you set off explosions underground to basically get oil and gas. And the special fluids that are used to pump that material back, and that material is very high in salt. And the US EPA, around about 2008 or 2010, became very concerned about salt pollution because there's no real regulations for it. And so what they did is they proposed a criterion based simply on measuring conductivity in the water. And that was about close to 10 years ago. Now, if you look at the major ions that we worry about for mixed salt toxicity, they vary a lot in terms of how toxic they are. So sodium and chloride, we eat this stuff. It's not very toxic at all. Sulfate is not very toxic. Calcium is not very toxic usually. In fact, it can protect against the toxicity of other salts. But as you know, potassium is very, very toxic. Magnesium is very toxic. It causes neuromuscular problems. And bicarbonate is really quite toxic because it disturbs the pH of the blood, makes the blood go alkalotic. Now, all of these contribute almost equally to conductivity, even though they have very different toxicity. And also, there's other proposed regulations based on what are called TDS, total dissolved solids, or simply the osmolality, or osmolarity. And that again says, let's just treat all of these salts the same, because they contribute about equally. But it seems unlikely that a regulatory approach based on treating all ions the same will be successful. In other words, it doesn't really make sense just to use conductivity, total dissolved solids, or osmolality alone. Nevertheless, the US EPA really needed to do something. They were under pressure to do something. And eventually they proposed a conductivity-based criterion. And this was published in Environmental Toxicology and Chemistry in 2013. It has not been implemented, but it's out there for what we call public comment. And there's been a lot of opposition to this. And the number that they specified, which would be protective, they thought, for 95% of aquatic life, would be a conductivity of 300 microsiemens per centimeter. Now that probably doesn't mean anything to you, but if you would just say that was all due to sodium chloride, that would be 3.2 millimoles per liter of sodium chloride. That doesn't seem like very much sodium chloride. Now, industry doesn't like this. And I think they have some good reasons. And one reason is it doesn't make much sense, this number. Now, if you're involved in toxicology, you know that if you want to get your toxicity tests accepted by the US EPA, you have to perform them in what they call US EPA moderately hard water. That's a special recipe that the US EPA gives you to make up the water. And this water is said to be representative of the water across the United States. It's kind of an average water for all the United States. When you make up this water, it says that when you make it up, the conductivity should come up between 275 and 325 microsiemens per centimeter. So what they're saying is the water that they think represents the background water in the United States would be in violation. It makes no sense at all if you think about it. But this often happens in toxicology when one set of scientists don't talk to another set of scientists. The ones who came up with this criterion were working in the Appalachian Mountains in eastern United States, where the water is very soft. And the organisms there are adapted to very soft water, like the Amazonian organisms, like the Rio Negro organisms. So that might be a very good number for the Amazon, or it might be a very good number for the Appalachian Mountains, but it makes no sense at all as a national standard. So, there's lots of public comment, a lot of opposition to this proposal. And in 2014, an organization called the Electric Power Research Institute, which is a collaboration of all the electricity generating companies in North America, decided to lead the fight against this new regulation. And they didn't like it for two reasons. One, the number made no sense as a national standard, and secondly, it didn't distinguish between the toxicity of different types of salt. And so they convened a workshop to which myself and Kevin Bricks were invited in 2014. And we were charged with asking this question: is there a better approach based on physiology? Now you might wonder why a research organization like this would be interested in physiology rather than just straight toxicology. But I think the reason is that physiology had been at the basis of this model, which I know some people in this lab have worked on, called the biotic ligand model, or BLM. And this model was developed over a period of about 15 or 20 years. My lab was heavily involved. Kevin Bricks was heavily involved. Dal's lab was involved. Labs all over the world were involved in feeding data into this model. And it's proved incredibly successful. It's the basis of regulations now for metals in the United States, throughout Europe, in Australia, New Zealand, some South American countries, China, and it's now being implemented in Canada. And it's a model which is based on a lot of geochemistry here, which I won't go into, but it's also based on the physiology of the organism. And that's where the contributions of my lab and labs like Dal's come into it. And it puts together an understanding of geochemistry and physiology, together with a lot of toxicity testing, to come up with regulatory needs. What I mean by that is regulations. And it makes a regulation which is specific for a particular region based on the background water. So it's very specific, this. Now, the reason why it's been so successful, I think, is that this biotic ligand model predicts a single parameter, the metal burden on the gill. It just predicts one thing. And if that prediction is right, we can relate that directly back to toxicology and regulatory needs. But it takes into account the physiology of the organism and all the background chemistry in the water to come up with that one number. The model is a big computer program. It's quite complicated, quite difficult. But the concept is very simple, and that's why it's proved successful and is now used as the basis of regulations. And this was published by a fellow called Paul Paquin and 21 others. I was fortunate to be somewhere in the middle of those 21 on that paper, as was Kevin Bricks, back in 2002, in Comp Biochem Physiology. We don't normally think of this as being the best journal in the world, but this, in fact, has had an immense impact. It's the most downloaded paper, the most cited paper this journal has ever had. Okay, that's a bit of background. So, they wanted us to come up with something like the biotic ligand model for salt toxicity. And they wanted some single parameter that we could predict with the model which would relate to toxicity. So we presented them with two ideas at this workshop. One was called the SID approach, which means strong ion difference. I'm not going to say too much about this. The strong ion difference is the difference between all of the strong cations like sodium and calcium versus the strong anions such as chloride and sulfate in the blood plasma. And if you disturb that, you end up with a fatal acid-base disturbance. And therefore what we thought is maybe we could figure that out and use it to predict blood plasma pH as the single thing that we would predict in the model. And the other idea we gave to them was an electrochemical approach. And that was that there'd be a disturbance of the transepithelial potential, or TEP, across the gills, associated with fatal ion regulatory disturbance. In that case, we predict this gill transepithelial potential. And they said, well, we kind of like those ideas. They're pretty nice. Thanks for coming. But we really like this one better. We're going to work on both of them, but this is the one we really like right now. And the reason we like it is that we've already hired a consultant who's come up with something rather similar to this. And we're glad you guys like it as well, because it means that he's probably on the right track. Well, this consultant turned out to be this guy, Paul Paquin, the guy who wrote that fundamental paper on the biotic ligand model. Now this guy, he was calling it at that time a membrane potential model. This is back in 2014. And I must say, this is a guy that I really have a lot of admiration for. He's an engineer. He has no background in biology or physiology, and anything about biology and physiology he's taught himself. But he's most engineers I don't think are all that smart. But this guy is really, really smart. And he really gets mechanistic physiology and toxicology. And every so often I'll get a phone call from Paul asking me some really crazy question until I realize it's a pretty good question you're asking, Anyway, he came up with the membrane potential model. And so they said, well, since Paul's working on this membrane potential model, it seems like your idea. We're not going to give you any money, but, thanks for coming and talking to us about this. So, just to get into this a little bit, I think you have to understand a little bit about the ion homeostasis in freshwater animals. Some of you know a lot about this, some not very much, so this is just kind of a little bit of a primer. But a typical freshwater fish, which for me in Canada would be a rainbow trout, lives in fresh water where the ion concentrations are really pretty low. These are the major ions, and most of them are a lot below the level of the blood plasma ions. So you can see, for example, that sodium and chloride is up to 150 millimolar, and the fresh water is down just a couple of millimolar. And the calcium's higher and the potassium's higher. Magnesium and sulfate are sometimes pretty close to the water. The bicarbonate is higher. But the animals typically maintain their internal ions very constant at levels far above those found in the dilute fresh water. The problem they have is they're always losing ions by diffusion across the gills. They leak out. So the fish have on their gills active sodium, chloride, calcium, and potassium uptake processes. Sort of things that, for example, Susanna's measures sodium, potassium, ATPase all the time. That powers those processes. And that maintains the homeostasis. So the gills are the main site of active ion and also acid-base regulation because they regulate bicarbonate, in addition to respiratory gas exchange. So the gills have two major functions, respiration and salt balance, or ion regulation. Now in this electrochemical approach, we have to think about the factors which would affect ion fluxes at the gills. Well, there's active transport, takes ATP. This passive permeability, that's the passive loss going on. And as a general rule, the permeability to cations, the positive ions like sodium, is always greater than the permeability to the negative ions like chloride in fish gills. So sodium is more permeable than chloride. There's also the big chemical or concentration gradients. The big difference in absolute concentration between the blood, which is high, and the water, which is low in concentration. And then there's the electrical gradient, which is the transepithelial potential. And I don't know, some of you probably see me measuring this in previous visits down here. I just do it the simple high impedance voltmeters. The electrical gradient and the chemical gradient together determine the electrochemical gradient across the gills. Now, just a kind of a note here. People often start to think about, oh, well, there must be more sodium leaving than chloride, and so there's a big disequilibrium of the cations and the anions, but that's impossible. Basic laws of physics say that there can never be any macroscopic charge imbalance in solutions, either in the exposure waters or the body fluids. This is theoretically impossible. The concentrations of cations and anions in a solution are always equal. Just keep that in mind. It might help you a little bit. Okay, so how do these things work at the gills? Well, this is kind of a model you've probably seen before, some of you, of the transport cells at the gills. And here's the dilute external fresh water. And here's the concentrated blood plasma. And the basic problem is the ions are leaking out between the cells. Sodium chloride leaking out. Now, just a kind of a point here, Paul Paquin's model was called a membrane potential model. And we had a lot of discussion with Paul before he convinced him that he wasn't really talking about membrane potential. The membrane potential is the charge difference between the cytoplasm and the water here, or between the cytoplasm here and the plasma. And that's not what we're talking about in this model. So he just had the terminology wrong. What we're talking about is the transepithelial potential, which is the charge difference between the blood and the external water. And in most typical fish in fresh water, it's around about minus 10 millivolts, something like that. Minus 15, minus 10, minus 5, it varies, but it's a number like that. So we got the problem, the ions are leaking out all the time. And we know now the mechanisms of active transport. Sodium moves in through a channel here, powered by proton ATPase, and also through a sodium-ammonium exchanger. We have recent evidence that potassium can go on that exchanger. Chloride moves to a chloride bicarbonate exchanger. Sulfate and bicarbonate can also go on that. Calcium moves in through a calcium channel, and magnesium probably moves into that channel, though we're not sure. And then there's these ATPases on the basolateral membrane which power the whole system. But if you get high levels of salt out here, the entry could simply just be by passive diffusion from the water into the animal. So that's the background. So in this electrochemical approach, it gets quite mathematical. I'm going to try to keep it at a simple level. But the electrochemical gradient across the gills is the arithmetic sum of the transepithelial potential and the Nernst potential. And some of you who've taken physics or cellular physiology have heard about the Nernst potential. It's the sodium level outside divided by the sodium level inside, the natural log thereof, with these constants here. We won't worry about those. So the idea is that sodium is very permeable at the gills, and you've got kind of basically just a sodium diffusion potential. Sodium is trying to diffuse out. It makes the inside negative, and that stops any more sodium moving out. And there's two components, electrical gradient and chemical gradient. Now, if the animal is at equilibrium, oh, by the way, we have to use activities, not concentrations here. Activity and concentration are just about the same in freshwater, but because there's a lot of protein in plasma, the activity of ions is reduced to about 75%. So at equilibrium, if you just rearrange that last equation, the TEP is going to equal the Nernst potential for sodium. And think of it, the TEP as being the difference between these two poles on a battery. Sodium is trying to diffuse out. Inside becomes negative as the sodium leaves. That stops any more sodium diffusing out. Now, I wish it were that simple. I think you can eventually get your head around that, but it gets more complicated, unfortunately. I'm just saying here, the TEP in freshwater animals is generally considered to be as largely a sodium diffusion potential due to higher permeability to sodium than to chloride. But see, we haven't even put chloride into this equation. We have to. We have to put lots of other things in. So to do that, we have to write another equation, which is like the Nernst equation, but it will consider every cation and every anion. So here you have the external cation. This could be sodium, and this could be sodium here. And you could add another one here. Or if it's an anion, you have to reverse the order. So the inside goes on the top, and the outside goes on the bottom. And you can just keep writing this for all of the single charged anions and cations. And this is pretty neat stuff, because these guys, Goldman, Hodgkin, and Katz, they got the Nobel Prize for this equation back about 1940. So now what we see is that the cation permeability is greater than the anion permeability, and all of these are going to have some role in the potential. Now, it'd be nice if it was that simple, but in fact, this only works for ions with single charges. If you have some ions with double charges, like calcium and magnesium, you have to write another equation, the Spangler expression. Now, I thought maybe I'd show you the Spangler expression, but then again I thought, no, this would be your reaction. So let's not worry about this. And it would take me like half a day to just type it all in here. It would fill that entire screen. Take my word for it. It's like the Goldman equation, but it kind of works. It works. Okay. Back to Paul Paquin now. He beavered away at this, and he's a modeler. He doesn't do any experiments. He just models data. And by 2016, he'd come up with what he called at this point now the EPRI mixed ion toxicity model. He's no longer calling it a membrane potential model. He wasn't calling it a TEP model. He's calling it a mixed ion toxicity model. And he based it on the Goldman equation and the Spangler expression. And he came up with a model which uses literature LC50 values, that concentration which is acutely toxic to 50% of the organisms. He used literature values for the internal ion concentrations. And then he fitted the permeability coefficients. And I should go back and explain that here, these PCs, these are permeability coefficients. So you have to weight each ion for its permeability. And he just fitted whatever permeability coefficient would work to explain the toxicity data. And sometimes he used interaction terms. But he came up with some model which seemed to perfectly describe a whole lot of data in the toxicology literature. And he had access to the US EPA database, which is run by a fellow called Dave Mount at US EPA, who's a collaborator. And this is one of the examples. Here's for the juvenile fathead minnow. And he predicted the TEP. And basically he said that if the TEP is minus 6.2 millivolts, 50% survival will occur. And if it's greater than that, say up here, the animals will all die. And if it's less than that, you get 100% survival. So this is many, many different types of salts here, all fed through this new model. And when he showed us this, we were kind of astounded. This is another workshop that Kevin and myself were at. And we were frankly very skeptical, because the TEP was modeled, it had never been measured, the internal concentrations were assumed values, the permeabilities were fitted to the toxicity data, and the model was based on absolutely no physiological measurements. No proof of principle experiments had been performed to validate the model. But nevertheless, we knew Paul was a pretty smart guy. So we thought he's onto something. So we said, we know how to measure TEP. Why don't you give us some money and we'll check the model now? Well, it's 2016. And EPRI basically didn't want to gamble. I think they didn't want to run the risk that their model might not work. And they also knew the politics was changing in the States to a new government. One government would not be worried about salt toxicity while they're in office, Donald Trump again. So they said, no, but we're really interested, but no, we're not going to fund you. But another organization, Rio Tinto, and this was at the time that the head of research was a guy called Bill Adams, said, I think you guys are onto something. I'll fund a pilot study. So he just gave us a few $1,000s for 2 years to do a pilot study, and off we went. So we know how to measure the transepithelial potential. And what you do is you take a fish, you perform an operation on it when it's under anesthesia, and you implant a catheter either into a blood vessel or into the body cavity. And it turns out it works just as well to go into the body cavity as it does into a blood vessel. We validated that. Then you allow it to recover overnight. And then you hook 2 electrodes. There's an electrode there and an electrode there to the catheter and to the water. And you put, these are silver chloride electrodes with 4% agar, 3 molar KCl bridges. And you express the transepithelial potential as the blood side inside relative to the water side. So it's always the inside relative to the water you're measuring. And you have to do overnight recovery from surgery prior to measurements. And again, just think about, like measuring the potential between 2 poles of a battery. So we decided to do a survey of 3 species. We did the fathead minnow because the US EPA really cares about this. It's used in regulations. But this is a tough animal to work on. It's very fragile. But we like the channel catfish because it's a very resistant animal. And so we thought, well, this seems to have the same mechanisms as the fathead minnow, but it's much tougher. Let's work on this. And then we also worked on bluegill sunfish because they have an entirely different gill ion transport system, which I won't go into, just to see if there was any generality in the results. And we had 2 6-week sessions down in Miami in the spring. I generally go to Miami in the spring and work in the lab there, and I worked with Kevin Bricks in the lab for 2 springs. Over those 2 springs, we made more than 9,000 measurements of TEP manually, which is I made all the measurements, Kevin made all the solutions. It's incredibly tedious, but you get the data right away. And it's always satisfying when you get data right away. That just shows, this is after the end of the experiment, after the fish had been euthanized. This was a channel catfish. Now the first thing we learned is that TEP was negative, but it's very variable, both in individual animals and amongst the 3 species. This is the fathead, this is the catfish, and this is the bluegill. So we wrestled a bit with how to deal with this variability, and we decided that we would therefore express all the data as delta TEP, the change in TEP against the background water, which was US EPA moderately hard water. So this is what we got. So this is for just sodium chloride as the toxicant. And we're measuring delta TEP. Now we had LC50 data from the US EPA only for the fathead minnow at this time. This was published by Dave Mount back in 1997, a very extensive study on single salts. So he based everything on the toxicity of the fathead minnow. So that point there is 4 times the LC50 concentration. This is 2 times. This is the LC50. That's 0.5. This is 0.25, etc. So it's a geometric series. And what we generally got were these rather satisfying hyperbolic relationships of increase in TEP, with the fathead always being the most sensitive and the bluegill always being the least sensitive and the channel catfish being in between. And notice how this tends to flatten out at around the LC50 concentration, which was interesting. That's the LC50. So we did 8 salts in Miami, 8 different salts. 6 of them are shown here. And we've actually done 2 more now back at UBC. And in all of them, you get these rather nice relationships. Sometimes it's not so great for the bluegill, but for the other 2 species, you always get these rather nice hyperbolic relationships. And they always flatten out right about the LC50 for the different salts. Now, look at these LC50s. There's potassium sulfate. That's really toxic. The LC50 is 5 millimolar. But look at, for example, sodium chloride. The LC50 is about 110 millimolar. Not very toxic at all. And yet, the relationships always flatten out around the LC50, which frankly really surprised us, you know. So we did 8 salts in Miami, and we looked at the delta TEP at the LC50. And it ranged from about 6 up to 9 millivolts. So it's very consistent, even though the concentrations causing this are very different. So there's remarkable consistency of the delta TEP at the LC50. There's a 1.5-fold variation in the delta TEP at the LC50, even though there's 22-fold variation in the salt concentration. So now we're starting to think, yes, Paul Paquin has basically got it right, and you can build a model on this. If you predict the delta TEP that's causing toxicity, you build your model on that. So we looked at the other two species, and it worked really quite well as well in the channel catfish. And with this one exception for KCl we don't understand, it works pretty well in the bluegill sunfish. Again, reducing the variation. And we wondered, is this stable over time? I mean, we're just doing, these experiments only take about 90 minutes to get that whole, for one fish it takes about 90 minutes to get that curve. We do generally 6 to 8 fish to get a mean. But that's all done very quickly. So we wondered, okay, what about if we wait a while? Are those TEP changes, are they stable? And we can only keep the catheters going for 24 hours, but there's no significant variation at all in any of these, including the control over 24 hours. So this change in TEP, and this was done at less than 50% of the LC50. It was done at 50% of the LC50. Is stable over time. No significant changes. We'd like to go longer, but we haven't done that yet. And we also wondered, what about an osmotic effect? Because, another idea for regulation is that it's an osmotic problem the fish is having. So we took the osmotic concentration up to 900 milliosmoles with mannitol. And that's the highest salt concentration we'd ever used. It had no effect at all on the TEP. So osmotic pressure or osmolality has nothing to do with this. So no significant effects of osmolality in fish. We do believe in Daphnia this is an issue, but not in fish. Well, another way of taking that same data I showed you is to plot it as percent of the LC50. And you can see that all the curves expressed as percent of LC50 really collapse basically into one curve. So that's the LC50, and these are various percentages of the LC50. And we're at another workshop where Dave Mount, our US EPA colleague, was there. And he said, you guys are onto something, but you don't have any sensitivity here. Like, there's the LC50, and the delta TEP at half the LC50 is just about the same. And even at 25% of the LC50, it's just about the same. Your curve is very flat here. So is this really going to be very useful, this indicator? But then he pointed out, what about using an index down here where there's a very steep part of the curve? And that I thought was pretty clever. So the problem is this delta TEP at the LC50 is uniform, but has no sensitivity due to the asymptotic or flat nature up here. But that was consistent with the APRI model, which had a very steep dose response. So he suggested we think about down here. So what we came up with is an idea of delta TEP EC50 as a toxic unit for salt concentration. So the idea here would be figure out what concentration of a salt causes 50% of the lethal delta TEP. Causes 50% of the lethal delta TEP. So that's going to be quite a lower concentration. And let's see if we can relate that to toxicity. So Dave Mount, by that time, had done a lot more testing, and he had data for all three of our species, which was great. And now we're using this new index, the salt concentration at delta TEP EC50, our toxic unit, against the salt LC50. And we get a very, very strong relationship for all three species. So this looks to be, I think, something that we can work with. Strong correlation in this more sensitive range. So we went back to EPRI again and said, look, I think we're really onto something now. And it's now 2018. They're probably getting a little nervous about the upcoming election as well. And they said, okay, yeah, we'll fund you for 2 years now. So we got funded now to really do this. And so that's paying for Bev Poe as a postdoc to do this work. And so far, EPRI have been just great with us. They're really good managers of our research for this. And so what we're trying is to do mixtures now. And we're extending this to other species, such as rainbow trout, such as daphnia, such as mussels, such as goldfish, a big range of different species. And we're starting to work with mixtures. And this is very complicated, very tedious, because you have, in this example here, we're using our toxic unit concept. We're making up 9 different concentrations of mixtures. And in terms of toxic units, at one end of the matrix, it's all of one salt, and the other end of the matrix, it's all of the other salt. And then in between, we use different ratios of the 2 salts. So now trying to do 2 salt mixtures. And I'm going to show you the best result we have so far. We have some results that are not so easy to understand, but this turned out to be our best result so far. And that's for the toxicity of magnesium sulfate versus sodium sulfate. And using the toxic units, this is what we get. We get this rather nice relationship, but we have this one outlier we don't understand yet. But at the same time, Dave Mount at US EPA has been doing the toxicity test to go along with this. This is classic concentration addition, and that's what US EPA sees when they do LC50 testing. So this is called an isobologram, and you can get different patterns on this. This would be for simple concentration addition. If you have an independent action, it would look like this. This is for concentration addition. So these 2 salts, both in terms of lethality and in terms of this delta TEP toxic unit, are acting by concentration addition, which is really very satisfying. So that's about the point we're at. We've really only got into the mixtures very recently. We've done another mixture that doesn't work out so clearly, which I'm not showing you. We have to work on that yet. So just end by thanking everybody who supported this, Rio Tinto, EPRI, Ecotox, which is Kevin's consulting company, and the national funding agency in Canada.